Bismillahirrahmanirrahim. Assalamualaikum and a good day to everyone. I would like to introduce myself first. I am Dr. Nur Hayati Hussein and my team would like to present on the topic characterization of three different types of pineapple waste powders. Okay, we are from Faculty of Food Science and Technology, Department of Food Technology, University Putra, Malaysia, presenting in this conference, International Congress on Biotechnology and Food Science. As an introduction, I would like to introduce you pineapple, or scientific name Ananas cosmosus, is a tropical fruit produced by Malaysia for domestic and export market activity. Malaysia is one of the country's pineapple planters, with a total area of over 6,000 hectares, and in 2012 produces more than 400,000 metric tons of pineapple. There are 9 major pineapple varieties planted in Malaysia, namely Morris N36, Josephine Sarawak Morris Ganja, Gando Yankee Josephine, and MD2. Okay, studies by Amir et al. Stated that N36 and Josephine were grown locally for the local fresh fruit market and not grown outside of Malaysia. And basically, we all know that commercially, pineapple fruit has been traded in the world in 3 different forms, which are in fresh fruit forms or canned or as fruit juice. Malaysia is also ranked 9th in the world, okay, in the world of pineapple growing position. There's a lot of pineapple waste was found, and this leads to environmental problems in Malaysia food industries. Production of pineapple is increasing day by day, thus increase the waste from the pineapple itself. Based on the previous study by Saraswati and team, who found that the pineapple waste contains antioxidant, sugar, phenolic compound, high in fiber, protein after maceration in ethanol. Another study by Sai et al. Stated that pineapple waste powder shows an increase in probiotic populations in yogurt as a consequences of pineapple powder supplementations increased from 0.3 to 1.4 log cycle. Whereas in another study suggested to use food powder with a moisture content ranges from 10 to 12% in order to slow the degradation process of the product. We move on to a table. In this next slide, table 1 shows varieties of pineapples planted in Malaysia. Another study who reported that N36 has its own characteristic, which the peel was in green color even though it was ripened. The difference between these varieties is based on the pineapple's shape, color, and taste. The different planting methods were one of the influences on the differences between varieties. It shows that Josephine and N36 has a cylinder shape, but Morris has a cone shape, different a bit. Okay, the peel, I share the design. Okay, here, if you can have a look. The peel is outside, okay, the outer layer of the pineapple. Okay, this is the peel. And while the core is at the inner part of the pineapple. Once you cut it, you will see the core part. Okay. The pineapples were selected based on the uniformity and shape, and also based on their commercial maturity stage. Okay, later I will show you which stage that normally used, and we use it as a reference for our sample too. Okay, Josephine is referring to a hybrid pineapple names from Johor, Spanish, and Sarawak Cayenne pineapple. Okay, this hybrid pineapple was introduced by Mardi, a local research center, in the year of 1996. Josephine is very suitable for eating fresh because it has a very good taste and aroma, with its sweetness between 16 to 18%. So natural sweetness taste makes this pineapple has a very high demand from Malaysians and has grown widely in Johor, Selangor, and Sarawak. Okay, Morris pineapples are also known as Ananas Mauritius. This type of pineapple is easily found in the market or supermarket in Malaysia because of their characteristic that it's very easy to plant and can grow rapidly in the open space, and it has been planted by 13,750 trees per acre, making it one of the commercial pineapples in Malaysia. Morris has a very unique taste, even though are not ripened yet, but the flesh of the fruits have a very fresh taste. The composition of this cultivar is high in sugar, ranging about 16 to 17% grapes, thus suitable to consume fresh rather than for canning purpose. Okay. Other than eaten fresh, this fruit usually used to make raw pickles, juice, and pineapple jam. Okay, thus Morris pineapple has the highest demand from customers compared to other types of pineapples. Even an international tropical fruit network claims that Morris has conical shape, deep yellow color, and deep eyes, which require a thick cut to remove the peel completely. Thus, this contributes much waste to the environment. So this is why we select this study too. Furthermore, N36 is only planted in one large area with 6,000 acres at Johor. This variety is mainly for canned products. Okay. Another study stated that N36 is the only type of pineapple that has high resistance to black heart disease problem. So the disease is caused by storage condition at low temperature. Thus, N36 has earned the biggest stake of as the biggest stake of the pineapple market in Malaysia, and this will lead to waste management too. Okay, so next is slide for problem statement. So study stated that half of the whole weight of pineapple was used for consumption and further processing, while another 20% consists of waste. So the waste part include core, stump, peel, and crown. Um, Another reason is that none has studied on the development of waste powders from local pineapple and compare the different types and parts after dried at different temperatures. Right? Um, we go next to my objectives. Okay, so it's actually to determine the effects of different drying temperatures on total phenolic compounds of peel. So we use peel and core of three different types of pineapple. So in this study we use three types: Josephine, Morris, and N86. And secondly, to characterize powders developed from the different parts of waste of local pineapples after drying at selected temperatures. Next, we go to methods. Okay, sample preparation. Okay, again, we just highlight the three types of pineapple used in this study. Okay, as you can see there, we use color number 4. Okay, um, to select for further study. Okay, because of their uniformity and again their commercial stage, mature stage. Okay, so all these sample purchased locally. Okay, the thick layer of the skin was peeled off from the fruits manually using a sharp knife. Okay, in order to minimize cross contamination from foreign matters. Um, During processing, the crown of the pineapple was discarded and the pineapple peels were washed thoroughly under running tap water to get rid of impurities on the surfaces. Okay, the preliminary test was conducted to try all pineapple waste, peel and core using an oven. We use three different temperatures, 50, 70, and 90, okay, at different durations. You see 50 degree for 12 hours and 90 degree with the lowest duration. And then we go for grinding using mechanical grinder, okay, in order to have a moisture content of 12 to 14% to get a very good powder. Okay, therefore we choose that particular temperature and that particular moisture range. Then we save it using 14 mesh size and store the powder in dark plastic container before we proceed to the listed analysis. Before analysis, we keep it in a chiller. Okay. Then, if you can have a look at the next slide. Okay, it's on the preliminary results of the TPC for the peel and core. Okay, you can have a look at 90 degree especially highlighted there that especially N36 will have significant difference if compared with 50 degree and other parts of the pineapples itself then, among the three pineapples then. Okay, therefore we select 90 degree and on top of that because it's very short duration, just 7 hours as I've mentioned previously. And we found out that yes, at 90 degree, Josephine and N36, as shown here, has a very good results of TPC as well as the core for the N36, right? Move on to we move on to TPC, which has been significantly affected by the different parts and drying temperatures that we use in this study. So again, 90 degree shows significantly highest TPC compared to the two temperature. So 90 degree was selected, and we can see Josephine peel pineapple powder has the highest TPC than its core powder compared to others. All right, Kim and Chin 2016 studied on the effect of drying temperatures but on different sample tomato powder, and they found that high drying temperatures affected significantly higher content of TPC compared to low drying temperature 60. And however, the air drying at 80 degrees increases the number of free phenol groups due to the hydrolysis of flavonoid glycosides and or release of cell wall phenolics. On top of that, also heat may provide energy to break the linkage between phenolic and the insoluble polyester that potentially increase the polyphenol bioaccessibility. Another study in 2014 stated that different TPC value might be due to the different initial TPC in the species or cultivars or even the geographic region of the pineapples. We move on to next slide, showing the proximate analysis for both peel and core of the different types of pineapples. Okay, you can have a look at the specially highlighted here, protein and the fiber of N36, which has good results. Okay, um, Morris pineapple has a significant content compared to others. For Bronze 2017 stated that amount of ash content in pineapple peel from Brazil was 5% higher than Morris peel found in this study. It stated that the ash composition indicates the variation of mineral elements in the pineapples. Another study stated in 2011, composition of fruits content depends on type of soil used on the plants, water used for irrigation, and types of fertilizers. Okay, in this study, N36, peel and core significantly has higher moisture content compared to others, especially Morris. Okay, meaning that high amount of moisture will give more time for drying process. So this is difficult, limited. I mean the limitations of drying the N36 then. Okay, as we all know, pineapple has a very good source of protein, namely bromelain. Although it has very low protein content, but it has bromelain with protease activity used in food industry, which has good benefits. Okay, the protein type of we call it as bromelain. Significantly higher amount of fiber found in N36, okay, than in Josephine and Morris. For both waste part, peel and core. Another study in 2014 on peel fiber content of the dietary fiber were 1.1%, significantly lesser than found in pineapple used in our study. Okay. We go next to the particle size analysis. The powder itself for peel and core of N36 is bigger, okay, than if we compare with Josephine and Morris, okay, with the value of 212.46 micron and 554.08 micron respectively. Okay. Another study stated that on the effect of properties of pineapple dietary fiber, low fiber content will increase the fineness of powder particles related to low fiber content found in Josephine and Morris peel and core. Okay. Then we go next to water holding capacity and oil holding capacity, okay, for the different parts and the different types of pineapples, right? We have a look these functional characterizations of this analysis, a quite interesting finding, okay, which shows a bit of a significant difference. Okay, peel powders has significantly higher percentage of water holding capacity than core powders. Okay, so you have a look, peel and the core, right? So there's a significant difference there. Okay, um, Valley et al. 2018 stated that soy flour has the ability to hold water at 6.755%, which means that our study for pineapple has a bit higher percentage of value relating that WHC in pineapple pea powder is better than soy flour, right? Drying at high temperature during processing may slightly affect the water holding capacity in pineapple pea powder, supported by another study on soy powders, where heating at high temperature helped to unfold and denature the protein structure and expose the side chains. Combined water does help in increasing the water holding capacity performance. The core powder has low ability to absorb water due to the low protein content. Moving on to next slide. Um, and comparing antacid with the other pill, the water or oil holding capacity is higher than compared to the wheat flour, 1.12%. Okay, another study stated that oil absorption ability of food material depends on the type and content of hydrophobic fractions present in the matrix structure. The presence of hydrophobic amino acid in the structure of a powder sample responsible for oil absorptions. Okay, the ability of N36 to hold oil is low compared to dried durian seed gums, which has better value than pineapple powders. Okay, might be due to less amount of protein content and zero amount of fat in the pineapple peel and core, as we mentioned in the table previously. Okay, Thibaudin et al. The presence of fiber in pineapple peel and core influence the ability of water holding capacity and oil holding capacity in the powders. This is the result for salt water solubility index, peel and core powders again. Okay, just sharing of the finding for N36. Okay, it has quite an effect. Okay, um, so solubility of N36 pill 60.55% was significantly higher compared to Morris pill powder, and there was a significant difference between Josephine core and Morris core. It indicates that the core powders have the higher ability to solubilize in water. Okay, meaning that it has higher percentage of water solubility. Okay, all the pineapple powders used in this study have moisture content between 10 to 12%, related to another finding, okay, which shows that low moisture content of powders will increase the water solubility index. Okay, another study stated that the solubility of powder depends on the moisture content, which high moisture content ranging 15 to 38% will decrease the performance of solubility. Then the solubility of the powdered sample is essential as the rehydration process. So it's very important for us to test this index. Will occur when the dried pineapple powder comes into contact with water. So a good rehydration process will wet the dried powders quickly and it will dissolve without float in the solution. Okay. Another slide showing the color analysis, the L, A, and B value for peel and core, and the difference between the types of pineapples. Okay, with as you can see the highlighted figures here, the N26 with a lighter color, very much lighter. Okay, compared to the others, Josephine has a different color of red and yellow for the peel and core. All right. The N, as I mentioned just now, okay, these findings are supported by another study due to another study found that the pineapple, as a pineapple trader, N36 pineapple, the color of the flesh and core are naturally pale and yellow. Pale yellow and more to white color, even though it was ripened and reached to the maturity stage. Okay, for Josephine Peel has more red than N36. Drying at 90 degree is believed affected the color of the powders after it was dried. Changes in color parameters during drying depends on the temperature of drying air, where high air drying temperature will lead to more visible color changes. Okay, the core of the Josephine has significantly higher value on yellowness. Strongly depends again on drying temperature too. So for the conclusions, we hope that these may exist in the country to reduce waste, where N36 pineapple powder is dried at 90 degree. Okay, the selected few drying temperatures may be considered as a functional food ingredient as it consists of higher fiber content, better water holding capacity and solubility, with paler color than Josephine and Morris. The powders may have the potential to be applied as food additives in food products. These are my references, and of course, you may contact me at this email, okay, ariatihusin at upm.edu, and don't mind. You are welcome to point any question if you don't understand any of it. Thank you so much, and hope this may benefits um, to the listener. Thank you so much again, and have a nice day to all of you. Thank you.